Good morning, sir. My name is Rosalinda Marie B. Ballesteros, and I am going to discuss the process of mitosis and meiosis. First is, what is mitosis? Mitosis is a type of cell division in which one cell, called the mother, divides and to produce two new cells called daughters that are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of the cell's nucleus is split into two equal sets of chromosomes. Next is the phases of mitosis. First, early prophase. The mitotic spindles start to form, the chromosomes start to condense, and the nucleolus disappears. The chromosomes start to condense, making them easier to pull apart later on. The mitotic spindle begins to form. The spindle is a structure made of microtubules, strong fibers that are part of the cell's skeleton. Its job it is organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The nucleus or nucleoli, plural, a part of nucleolus where ribosomes are made of, disappears. Next, late prophase. For metaphase, the nuclear envelope breaks down and the chromosomes are fully condensed. In late prophase, sometimes also called prometaphase, the mitotic spindle begins to capture and organize the chromosomes. Next is metaphase. Metaphase chromosomes line up at the metaphase plate. Under tension from the mitotic spindle, the two sister chromatids of each chromosomes are captured by microtubules from opposite spindle to poles. Next is anaphase. The sister chromatids separates from one another and are pulled towards opposite poles of the cell. The microtubules that are not attached to chromosomes push the two poles of the spindle apart, while the kinetochore microtubules pull the chromosomes towards the poles. Next is Tylopase. The spindle disappears. A nuclear membrane reforms around each sets of chromosomes, and the nucleolus reappear in each nucleus. The chromosomes also start to decondense. Next is, what is meiosis? Meiosis in human is a division process that takes us from a diploid cell, one with two sets of chromosomes, to haploid cells, ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and an egg join in the fertilization, the two haploid sets of chromosomes from a complete diploid set, a new genome. Next is phases of meiosis. Next is phases of meiosis. Meiosis I, a first round of cell division. Meiosis II, sister chromatids separate during a second round. The phases of meiosis I is prophase I, the starting cell in diploid 2n is equal to 4. Homologous chromosomes pair up the exchange of fragments in the process of crossing over. Next, metaphase I, homolog pairs line up at the metaphase plate. Next is anaphase. Homologous separate to opposite ends of the cell's sister chromatids stay together. Next is Tylopase I. Newly forming cells are haploid n is equal to 2. Each chromosome still has two sister chromatids, but the chromatids of each chromosome are no longer identical to each other. Next is phases of meiosis II. Prophase II, starting cells are the haploid cells made in meiosis I. Chromosomes condense. Metaphase II, chromosomes line up at the metaphase plate. Next is anaphase II. Sister chromatids separate to opposite ends of the cell. Next, telophase II. Newly forming gametes are haploid and each chromosome now has just one chromatid. And that's all. Thank you for watching.